Hi, welcome to Biology. Today we will be looking at B lymphocytes and humoral immunity. So in terms of the learning objectives, you need to be able to explain the role of B cells in humoral immunity, explain the roles of plasma cells and antibodies in the primary immune response, explain the roles of memory cells and antibodies in the secondary immune response, and explain how antigenetic variation affects the body's response to infection. So in terms of the specification, now we are looking at this bit here. So we need to look at the response of B cells, B lymphocytes to a foreign antigen. Remember, antigen is the foreign protein on the surface of the pathogen, and we need to be able to explain how B cells, how the plasma cells and memory cells will respond to this antigen. Okay, so a quick overview of what is the humoral immunity. So humoral immunity is the response that involves B cells, B lymphocytes. So it will involve production of antibodies. So it's a little bit different than cellular immunity with T cells. Here we've got antibodies, and antibodies are proteins. There are specific proteins which are soluble in the blood and tissue fluid. So in terms of the B cells, they produce a specific antibody. Specific antibody, that means that will have a specific 3D tertiary structure that responds to one specific antigen. So a foreign protein on the surface of the pathogen, and those antibodies are on the surface of B cells and will be complementary to the antigen. So antibody and antigen will make a complex. Okay, so antibody will bind to specific receptor which will be found on the surface of antigen. So that's why we say they will be complementary. An antigen will enter the B cells by endocytosis. Okay, and it will be presented on its surface. So B cell, as we've mentioned in previous videos, it's the antigen presenting cell. So how then T cells coming across here? Well, we need T helper cells. So T helper cells, specific T helper cells, will stimulate specific B cells to divide by mitosis. Okay, so mitosis, remember, it's a division where we are going to get two identical cells. Hence, we are getting a clone of identical B cell which will be able to produce antibody that is specific, complementary to this foreign antigen. Okay, this division of B cells dividing by mitosis we call clonal selection. Okay, and monoclonal antibodies, they are the same. So clones, mono means one, so clones of that specific antibody produced secreted by B cells. Right? So in terms of the specification, we've mentioned that B cells can develop into plasma cells and memory cells. Of course, there are differences between them. So where are differences coming from? Especially, we need to know which one of them is involved in the primary, which in the secondary immune response. So plasma cells, they are involved in the primary immune response, memory cells in the secondary immune response. How do they work then? So plasma cells, they will secrete antibodies straight into blood plasma, but in terms of the memory cells, they do not secrete antibodies directly, but they circulate in the blood and tissue fluid instead. In terms of the plasma cells, they produce large number of antibodies with a short lifespan, so that's really important. Those antibodies not going to have a long-term immunity, they're just going to be there for a short time. In terms of the memory cells, they will live longer and they will be involved in the long-term immunity. Right, so what is the job of the antibody then? Antibody leads to deconstruction of the antigen. Okay, but if we are talking about antibodies and memory cells, we need to be careful with this. We need to remember that memory cells are involved in the secondary immune response. So those memory cells will divide rapidly and develop into plasma cells to produce those antibodies. So every time you are talking about memory cells, you need to use the words like rapid, okay, and more antibodies compared to plasma cells. So plasma cells then will be involved in the immediate defense, and in terms of the memory cells, they will act faster than the primary response, so they kind of remember this antigen and have those complementary antibodies in place. Right, so let's have a look at quick difference here again between the primary and secondary response. And the graph shows you the density of antibodies in the blood against the time in terms of the first contact with the antigen. So in terms of the primary response, as we've mentioned, the number of antibodies produced by plasma cells will be lower compared to those produced by memory cells. Okay, so remember, memory cells will be involved when you are affected by the same antigen. Second time, primary response, plasma cells, when you are affected by the first time. Okay, so those are our differences. So what we've mentioned, primary response, this is when you first time are in contact with the antigen. Secondary, it's your second time. So primary, it's your initial. Secondary, of course, it's your second contact with the antigen. Primary works slower, secondary works faster, because already got this information in place about the complementary antibodies. Primary has a smaller response, which we've seen on our diagram, producing a lower number of antibodies, while the secondary is quite larger response and you've got a higher concentration of antibodies. What cells then are involved in the primary response? There will be T helper cells and B cells. In the secondary response, we're only looking at memory cells. So how does it work? Okay, so let's have a quick look at this diagram. So what we've got here, okay, here we've got a pathogen. On the surface of the pathogen, we've got antigen, so your foreign protein. Okay, here we've got the antigen presenting cell, so it's your B cell with the antigen displayed on its surface. So once the pathogen with the antigen engulfs, the antigen will be presented on the cell surface membrane of the B cell. Here, what we've got, then we've got the T helper cell that comes along and stimulates this specific B cell with the specific antigens to divide. How do they divide? They divide by process of mitosis. So once they divide by mitosis, they can develop into two types of the cells. So one cell is a plasma cell. So as you can see, plasma cell will secrete antibodies, and then here we've got a memory cell, which is already here with the antibodies complementary to the antigen, and those antibodies are on the surface of the memory cells. So you can pause the video in a second to make a notes to label your own diagram. Pathogen with the antigen on the surface engulfs. The B cell is your antigen presenting cell. Okay, so here we've got those antibodies which were secreted. Antigen is presenting on the B cell. B cell then it's activated by T helper cell to divide by mitosis, produces plasma cells, memory cells. Okay, plasma cells secrete antibodies, so they can attach to antigens on the pathogen later on in the primary response, and memory cells, they will be in the blood ready for the secondary response. Okay, so a quick activity for you here to list those events in the correct order, to explain, to describe how the B cells